Data from interviews with a meatball grinder proved that 55 of meatball sellers make rat's meat into their meatball dough. Meanwhile, the number of meatball sellers in Sumenep is estimated to reach up to 75 sellers, with 65% rat's meatball contain various bacteria such as Listeriosis, Salmonellosis, Leptospirosis, etc. As a predominantly Muslim country, rat's meatball are strongly opposed due to the prohibition of this animal meat. However, detecting the presence of rat meal in meatball still requires adequate technology and human resource. In the previous research by Baptiste 2022, ants have been trained for 10 minutes to identify cancer cells in rat urine. On the other hand, every food has volatile organic compounds, or VOCs, so both theories can be combined to create a new biosensor for rat meatball.
From the identified issue, we proudly present this research with the title Proliferation of Rat Meat in Bakso in Sumenep, utilizing local ants as a biodetector for the distinctive VOC of rat meat balls using Pavlov's theory and GCMS method and microbial testing. The objectives of this research are to determine the effectiveness of ants as a detection tool for rat meat balls and to identify the most effective ant species in detecting VOC of rat meat balls. The hypothesis of this research is that ant can differentiate VOC in rat meat balls.
First, ant preparation. Trap ants will be stored in the best variation of containers. Second, rat meatballs preparation. Capture rat will be cleaned and their meat will be chopped to create the mixture according to the proposed recipe. Third, classical conditioning adaptation. Each ant species is accustomed to the characteristic of rat meatballs with two variations by feeding them 3 times every 2 hours and 3 times in weeks. This is done in order to knowing the best classical conditioning method. The first conditioning includes sugar as a stimulus, while the second and third do not. Last is testing stage. This stage exposing ants to 7 different types of meatballs for 25 minutes with a randomized placement at the 15th, 20th, and 25th minutes. Then, software will be analyzed and behavior on this test.
After everything was completed, GCMS and microbial tests were conducted at the University of Brawijaya Malang Laboratory. The entire data sequence produced several findings that will be the main topic of this presentation. First, we will discuss the pre-treatment mechanism on ants. Ants are captured with sugar bait within 10 cm radius of the ant colony. After capture, ants are immediately identified by species by observing the body structure of ants using a microscope.
After identification, these 4 ant species are stored. However, during storage, many ants die. So researchers decided to use variation of containers. Variation 4 is the best result because each ant has different characteristics, and the storage of ants adjusts to the characteristic of the ants themselves.
Is the discussion of conditioning. Conditioning per day and per hour giving result in similar outcomes, as shown in the graph. The graph shows the number of ants that can survive during conditioning. This is correlating with ant capability. Referring to the updated data, and B is the ant with the best result in these two variations, and daily variation tend to have a lower result. In conclusion, hourly variations are more efficient for use. Testing on ants with this hourly conditioning will be carried out. During conditioning, it was found that ants tend to favor one type of meatballs, and A tends to approach these meatballs with 0 types approaching rat meatballs, and B tends to approach rat meatballs with an accumulation of 10 types approaching rat meatballs, and C doesn't tend to approach rat meatballs, doing so only 2 times. The same number means ants remain on rat meatballs. Based on the 3 graphs, it is concluded that ant B has the best memory, as seen in the 15 and 20-minute graphs, where ant B has the highest number of approaches to the rat meatballs.
Sample rat and beef meatball was tested in 2 variations, namely before and after cooking. Based on the result of the GCMS analysis, it is found that there is an increase in volatile organic compound or VOC content in both samples. This is proof that a material subjected to heating treatment can undergo changes in its chemical structure due to the heating process. The alteration in chemical structure occurs because of changes in bones, resulting in the formation of new compounds. Based on the graph of the GCMS test result, it can be seen that the compounds capable of reaching. However, the highest peak obtained doesn't become the characteristic of each type of meatball. Therefore, it is necessary to search the marker compounds through the identification of VIP values from each sample. The highest VIP value is differentiating each sample compound that remembered by ants. GCMS is conducted to determine the difference between VOCs in each sample. As the name, VOCs are volatile organic compounds, which ants are searched for food by detecting volatile compounds. Furthermore, microbial testing was conducted by counting the total take count, or TPC, of microbes in 7 meatballs that previously tested on ants. The test results were then compared with the meatball standards established by BPOM. As a result, all meatball samples met the criteria except for rat meatball. The BPOM standard for processed meatball product is less than 10 to the power of 5, while the microbial test result for rat meatball was 8.1 times 10 to the power of 6.
The GCMS test results show that there is a specific VOC present in red meat balls. Conditioning the ants has proven to detect VOC from red meat balls with the best result from N3 at the 10-minute mark.
Of ant conditioning is expected to be further developed, especially as a biodetector. Additionally, researchers hope that future research can be conducted to explore the utilization of ant as VOC component detector.